Okay, so today I'm going to walk you through how to image a slide on the Opera Phoenix. To start with, you're going to go to your setup tab and you're going to go over and just go down. I have one already loaded here, so that would normally be blank, but then you're just going to go and you're going to select which plate. And so the slide, there's a special PerkinElmer slide holder that's on the shelf above the Opera Phoenix. So I have the slide holder and the samples are below the slide. I want to image with the 5x air objective to do the pre-scan just to save. I just want to be able to find where my specimen is on the slide. I will then go back and image it on the 20x. So I'm going to just start with the 5x. You're going to hit add here to add your channel, and then confocal versus non-confocal you select here. Binning, the default is 2. To get help with any of these other things, to decide what you want, you can right click help. To channel, you're going to hit add. For mine, I just want DAPI, but there's a whole list of pre-loaded stains in here. So you're just going to select the ones that you want. You're going to hit OK, and then you can add as many as you want, or you can remove them here. If you have crosstalk in your samples that you find, you can select them here and say, I don't want my DAPI with whatever your other stain is, and it will separate them and image them separately. So then next you're going to go down, and for my slide, I want a little bit of an overlap. I'm going to do like 5%. I'm going to do -15. I'm going to do 15 slices, and I'm going to do them 5 microns apart. So this is going to go from -15 microns to 55 microns for a total height of 70 microns. You want to make sure that you use then test is checked. And then we're going to add an online job. So I already have one. I'll show you where to go and do that after. So I'm going to make sure that this hat says use and test. Now I'm just going to hit test. So on the right here, you're going to have I would have already selected the first slide. I selected a general area where I think the tissue might be on here. And then here's this shows my Z-stack that I defined on the left. And you can also hit enlarge, which will make this a lot bigger. It actually looks like my box moved on me, so we'll have to probably do this again. But then you can reduce that. Once you hit test to see your image, you're going to go over here to the image, and once this loads, you want to make this orange, and this orange. When it turns blue, that means that it has already taken the image. So sometimes you have to click on it again once it turns gray. So now you can go through each individual slice here. So you can see that there was nothing here. Maybe the very edge of it is here. So I'm going to go back and I'm going to do a bigger range. I think that my sample is somewhere down here. So I know it wasn't the center one. I'm going to deselect that. I'm going to do test again. So now it's going to image those 4 boxes. Back over here. So now I can see that my sample is here. This particular side has a cross section of a testis on it, so it's going to be a little circles. As I go through my Z-stack, I can see that this is in fact my sample. If I want to right click and do show intensity, and then you can use the mouse to zoom in or out, make sure it's on a place that has the stain that you want. So now I can go through and look at my intensity. Basically, you want the intensity to be a lot higher than this. And so this would be considered the background because your sample's not here, but your sample is going to be here. So you want to increase the DAPI. So this is at 60%. I'm going to bump this up to 80%, and I'm going to bump this up just a little as well. And I'm going to hit test again. The reason why I picked 80% is at this particular second, it says that I have this error that only 80% laser power can be used for DAPI. So normally you can go all the way up to 100%. Again, you just want to kind of pick a spot and you can look at what the intensity is. So this looks a lot nicer. The other thing to note is that there's 4 ways of looking at this. There's standard, enhanced, and highlight. And if I open this as I go through, so it's highlight, it's just basically changing these numbers, and you can manipulate them yourself. So, you know, if these spots are the only stuff that you think should have DAPI in it, you want to make sure when you're looking at your intensities that those are the spots that you're looking at. And again, you can zoom in to make sure that you're on one of those spots. And if you right click, zoom to fit, or you can right click and zoom this way as well. So for this tissue, because the cover slip is not completely flat, is the reason why we have a Z height, and we will continue to keep the Z height as we progress through. So this is about, the background staining is about half of like this particular spot. So there is a difference, which is important. So in this area down here, the DAPI stain is higher as well. So basically what we're going to see with the slides is they're going to be at different heights, and they're going to be, the tissue is going to be at different heights across. So what I do for this is going to be my analysis that gets attached to my pre-scan. So this analysis, I basically want to identify, this is where my testis is, this is where my testis isn't. So when you go back and image on 20X, I don't want to image this background area. I only want to image where my sample actually is. So for this scan, I'm going to do max projection. So I want all my Z slices here. So we're just going to select all of them. And as you can see, these overlapping boxes is that overlap percentage. So if you're not happy with the way that that's showing, you can increase or decrease your, overlap. So the next thing is you want this particular one uses the filter image. Let's see what is here. So as you can see, this particular protocol isn't very good. So I'm just going to go ahead and scrap this one. Let me just check real quick that I don't have one already made. So I'm going to pick, let's see if this one's any better. No, So that's the same one. Okay, so we're gonna start over here. So you're gonna go back here, max projection. This will show you if there's any flat field correction available. This one particularly doesn't have any available. I don't think this does anything because it's not bright field. So I'm going to try texture region and just see what this does. So I want my channels to be dappy, and there's no region of interest. So I'm going to split into classes. So my class A is going to be my testis. My class B is just going to be background. So what I do over here is I'm just going to go around and say, okay, this is the tissue that I want imaged. Just randomly select a whole bunch of it. And then red is going to be my background. So now I'm just going to put a bunch of spots on my background. And then I'm going to hit apply. So I can see that generally it's, the green is where my stuff is minus the center, which is, a fair thing to do. And I can go to another spot. You can see that it's, where there is tissue, it is pretty accurately detecting. So here, I would like this to do a little bit better. I'm also going to have it split into objects. Let's see if we can get this better. Just add a bunch of these spots to where the area is that doesn't have very good coverage. And I'm just going to try to see if I can eliminate any of these spots over here. And I see that this looks like it's probably some tissue over here, but ideally if I could get rid of that, we'll just see what happens. And then again, we'll do apply changes. So this area got better, this area also got worse. So now what I can do is, because I split it into objects, I can say, well, let's just try seeing what the intensity does, adding the intensity. So that made this area a little bit better, but what happens over here? So this is missing quite a bit up there. So let's see what happens without intensity. I think the intensity is variable, so so I'm going to come over here, and I'm just going to add some more. So this area, ideally would have some more of its more coverage. I'm just going to add here. And then there is no red on this one, so I can't actually do. You need both of these to show up on the same section. You need red and green dots. So this can be all get red dots. This is not the tissue that we want. Just go crazy, put red dots everywhere. And then green, put the green dots all over here where my testis is. And then do apply changes. If you go here, this is going to show you, like this one's invalid because I didn't have my class B on here as well. But if I hit apply down at the bottom, let's see how this looks. I'm not very sure where this is going. When I hit apply changes, it should just apply changes. Let's see. I have to start over. Okay, so let's reset. Okay, so again, just do a bunch of green spots. A bunch of red spots. Again, just add a bunch of green spots, especially to the areas I'm not completely happy with. And then do a bunch of red. Do apply changes, see how that looks. So I'm just gonna leave that for now and just see what happens. So the next thing I'm gonna do is I'm gonna calculate my morphology properties. So I want the area of my testis region. Testis region, and then I'm going to select a population. So I want my testis region, and I want by property. So I'm going to go to my area, and if you right click and highlight objects, it will tell you. So ideally, you're going to have a big area. So this one's 700,000. And you can click here to arrange by size. So I can say, okay, so this is like 1,500. Oh, it's the top. Now, these are my big. So let's just, use an arbitrary. I want it to be bigger than 400,000. And the better that you can get this to look, the more accurate this is going to be as well. For now, I'm just going to leave it like that. So I have my area. So now I want to determine my well layout. So I want to image my selected, testis selected population only. And I want to image that on the 20x field overlap. I think I want it to be slightly more. Let's just see what this says about field overlap. Default is 2. Can be increased if required, but should not be reduced to avoid gaps. So 2 is sufficient, they say. So we'll just leave it 3. And you can see, you can click on your different fields now, and you can see where it's going to image. So the other thing here is, say you don't want the entire thing imaged. On this particular one, the entire area is imaged and there is 24. So I'm just going to say I don't want anything more than 24. If you only selected and you only wanted some, or you only wanted, say, 5 from each part of the testis that's image, you can reduce this number. But for now, I'm going to leave it at 24 and just see what happens. So now you just want to make sure you click on results. Just and no results need to be reported, so you can just leave this as 0 and apply. This will go to 0 as well. This is all 0. So now you want to save this. So I'm going to call this 2020 8/18 testis pre-scan analysis. So let's do that. So now I'm going to go back over here to setup. Where I'm going to attach that is I'm going to attach that right here. So I'm going to hit this little 3-dot thing. You can organize by date, which is really helpful. So I can see there's the analysis I just created. So now that's there. So now I'm going to hit save. It's telling me I have nothing selected here. So I'm just going to select this. I have my little area that I know has cells. And now I'm going to actually select a different area just to double check myself. So I have this selected, I have my squares here, I have my Z height. I'm going to do a test. Because this is checked, it's going to also test my analysis as well. So as you can see, when this turns blue, that means it's fully imaged. And so basically for my Z height here for a slide, I just want to, remember this isn't your final image, but I just want to verify that the intensity is going increasing, but then also decreasing on the backside. So I want to make sure that across this entire slide, I'm going to get the brightest section. So this is done, so I can go over to my image analysis. I can pick one of these squares, select the Z height, select all my Z's. And now I can jump down to the bottom here and just see, okay, so this is where my testis is. This is where it's going to image. That looks good. Again, this is only imaging where my testis is. It's all testis. So I'm pretty happy with how this looks. It's not going to be perfect, but I'm pretty happy. So now I'm going to go back to setup. So now what I want to do is I'm going to save this. Let's just save this as today's date. Once I'm happy, and if I'm going to do more experiments, I can keep this as the same name. But for now, I'm going to keep changing it until I find a protocol I'm happy with. So now that's my pre-scan. I saved it. So now I'm just opening a prior rescan, and that's just because I forget what this is stained with. But again, you would hit new, you would start at the top. I have my slide holder. I want to do 20x water. I want to do confocal. My binning is going to be 2. I'm going to add my channels. So here I have Alexa 488, and I have my DAPI channel. And this is what I want imaged in my final image. So this is something that we're going to test, is my height. So I'm going to just start with -10. I'm going to do 23 slices, and they're going to be 2 microns apart for total height, and then it's going to go up to plus 34 microns. I'm going to make sure this is selected for use in task. I do want some overlap. It is defined in my rescan protocol, but just to be sure. And there's no online job here. You could set one up if you have later on, no, this is exactly what I want to look for. I personally like to go back and just run my own analysis. But so I'm going to select this. Nothing really needs to be selected here because your pre-scan is going to tell it what to do. Make sure my Z-stack looks good. And now I'm going to save this. So this is my rescan when I rescan on 20x. So now, because it's a pre-scan and a rescan, the only way to test if the rescan is working on an area that has my particular sample, I have to do the pre-scan and the rescan together. So I'm just selecting what I just created, my pre-scan, my rescan, and I'm just going to type test. So now this is going to run. And again, I can select this, and when this turns blue, that means it's done imaging. So now basically you can see, I'm just zooming in on my mouse, you can see that this is where it's detecting part of one testis and part of the second testis that's next to it. This particular slide has 4 testis kind of in a grid pattern. And this one that by default is clicked is going to be the first one that is imaged. So I'm just going to go to the bottom of my Z and wait for it to turn blue, which means it's already imaged. Because it's a new objective, the machine is now calibrating and doing this acquiring image at alignment parameters. It's going to take a couple more extra minutes to do that. So here is where the Z matters. I want to make sure that every one of these boxes has the highest intensity within that particular. Well, so you can look at these separated. If I just go back and redo that. So I can look at my intensity of my Alexa here and just to see, I'm out of focus here, you can kind of tell, but it's also a really low intensity. And then my intensity goes up, up, and then it starts going down. So this tells me that I have the right, my Z contains the right one. So say if I want to look at my little spermatid heads here, make sure I get it on the spermatid head. And just look at my, so ideally, and you can see that this is brighter over here at a different Z height than it is over here. And again, that's because the sample is not completely flat when it's on a slide and I'm looking at the cover slip. But I like to have my intensities above 2000 ideally if I can. And I want to have these, this Alexa above 2000 as well. This one's actually really high. I'm just going to look at, I like to look at my highlighted one as well, just to make sure that these are the cells that I am expecting to be highlighted here. I think these are supposed to be little dots, the little dots in these cells, I think is what I'm looking for this one. So I'm just going to double check. Yeah, see, so the maximum intensity I think is like 64 or 65,000. So I want to decrease my laser power for this particular staining. And again, this is image a whole bunch of them just to make sure that I'm in the right height for everything. So like now this one down here has these sperm highlighted. And as you can see that the host is a lot brighter in this part of the section. So I have to keep that in consideration too, because ideally I want my host to be bright enough, but not too bright in all of my sections. I'm just going to jump over here. So this particular section, I don't remember exactly which ones I'm looking for. This stain is on my nuclear head, so it's also in these cells. I want to say it's these cells that I'm looking at, and these are really high intensity. So I'm definitely going to back off my intensity a little bit here. And once you have enough, you can just stop it. I just wanted to see a wide variety. Yeah, so now I said, okay, my hoax, of course, these ones a lot brighter. But I still think I can increase my hoax a little bit and decrease my my other one. So I'm gonna go again to my rescan over here. I'm gonna go to my DAPI, which I think I want to increase a little bit. So I'm gonna increase the laser power. The laser power versus the time, the longer with the higher laser power that you use, the more likely it is to bleach your samples. So it's kind of a balance of increasing the laser power versus the time. The total imaging time is up here, is an estimate. So you can select just a random number. For this pre-scan and rescan, it's hard to know, but so this will give you an idea of how long this will take. So I'm going to start by increasing my laser power so I don't have to increase my time. Over here, this one was a little high, so I'm going to decrease my time over here. I'm going to hit save. This is going to ask me if I want to put this rescan protocol, and I want to update it on my rescan, which is over here. So I said, yes, please update this for me over here. So now I'm actually going to, so I saved that one. I'm going to go to my pre-scan, and I just want to look at a slightly different area over here. And I'm just going to real quick show you another thing that's really helpful. I think maybe I have another one up here, another test is up here, but I don't know for sure. So what I'm going to do is I'm going to select my area. I'm going to do my test. Click on my image, turn this orange, and I'm going to wait for this all to turn blue so I can look at my sample. Now I keep switching. Every time I do a pre-scan, it's on an air objective. Every time I do a rescan, it's on a water objective, which is another reason why I've been moving around. If you don't move to a different spot, the bottom of your slide is going to have a water droplet on it, and it's not going to be able to focus. So you want to keep that in mind. All you have to do is eject the slide, wipe the water off with one of these wipes that we have in here that are designed to wipe the slides, and then you're good to go, and you can put it back in. And then so you can go back and forth between the two. Just make sure you wipe the water off after each one. So this looks like I'm not in a testis here. So I'm just gonna put the next one. I'm not in my sample here either, or here, or here. So I'm going to go back to define layout. So I know I had samples somewhere closer down here. So maybe they're below the center. So I'm going to do the same thing. I'm going to select different squares. I'm going to hit test. And again, switch back to test images. Select this. So now I'm in my testis. So once this finishes, I'll show you how to save this image onto. So you go back to define layout and you select these wells. What do I do here? All right. So select your whole Z slice, select your squares, and then right click background for well. Please select one well plane time point only. Okay, so I can only have one plane. So I'm going to select one plane. I'm going to right click background for well. So now when I come over here and I enlarge it, this actually shows what where my sample is. So now if I want, I know the general pattern of my sample, so now I can say, okay, so there's probably testis here, one over here, one up here, one up here. And you can do this for the whole, you can do this for a whole area. So if you wanted to do this, find out which squares has your samples, and then you can go back and you can say, okay, I want to image only these four squares, because those are the only places that my sample is. So you can do that instead of using the pre-scan and the rescan. I have a lot of slides to image today, so I'm gonna keep it on the pre-scan and rescan, but this is another option. And it's just a little bit more involved. And the way to do that too is if you wanted to rescan on 20x, you'd have to go here to 20x water, and that stays, but your boxes change to the 20x. So now you can see how that changes. So I'm going to go back to my 5x. Okay, so I'm going to hit save here. Save everything. So now I'm going to do another test. So I know that I go here, I want to have only the area where there's actual testis. So I can actually do just one of these. So I'm going to just select this one region. I'm going to do save. So I'm going to go back to my run experiment. I'm going to call this test. And the reason is that for that is so that I can go back and delete all of the ones that I called tests, because I don't need to save this data. They're just tests and they're not my full sample. So I did my pre-scan and it came up with, this is where my testis sample actually is. Okay, so I'm going to select a spot on here. Just make sure that I have my brightest spot is in there. Again, I'm going to do the same thing here. And if I see that all my samples are all higher up here, I can actually change my Z-stack to delete this area down here. So these intensities are much lower than what they were before. And this kind of shows you the areas that have overlapping intensities. So I actually think it's these little guys here that we want to measure. Yeah, I think it's these little spots. So you can see that they're within these cells. So either way, I think, this generally these numbers are over 2,000. And once I get more involved in the analysis and stuff, I'll know what intensities are important. But the big thing is that they're different. They're significantly different than the background, which they are. And then you can see the lower stuff in here. So once you're happy with how this looks, you're happy with your Z height, you're happy with your intensities, you can go back and forth and keep rescanning and do a test. I'm going to start these at 0 and just take a little bit off. Maybe I'll even go up. Ideally, you want to test more than one slide. So if you increase that, then you can decrease this a little bit. I actually forget what I had this at, so I'm just going to go back and I'm going to reopen it one more time. Okay, so mine went to 34 microns. So I'm happy with this height. I just want to decrease this a little bit. So this is still around the 34. I might just do one more just to make sure. Okay. So now I have my height. Everything's what I wanted at. I have 2 slides in here. So you can see when I select this whole big area that it goes up to 5 hours and 15 minutes. But I'm pretty sure that my samples are all down in this. Oh, what I selected, okay, so this is a pre-scan, so that, or the rescan. So this, what I selected doesn't matter. So I'm gonna go ahead and save this with my 2 slides here. I'm going to do my pre-scan. I'm going to have my 2 slides here. Okay, so now I can select. So I think they're all in this general area, but if you don't know, you can select. I'll make it just a little bit bigger just to make sure I have everything there. And my whole test is there. And you can see that it's going to take 11 minutes to do the pre-scan, and that's going to be for both this section on both slides. So I'm going to hit save. I want to save this pre-scan to here. So now, one important thing is that you always want to include an assay layout. So I got to look up real quick what my samples are. This is just so that you can differentiate later on. These aren't my samples, so I'm going to put the information that I think is important here. But you can label, like these are some suggestions you can add. Anything that's numeric, only a number, alphanumeric, you can put numbers and non-numbers. This one also has a number on it. So you want to save that, save it as 2020 18, test this. You want to hit OK. So I'll load it to your screen. Yes, I do. Don't know why it didn't load. Oh, it's loaded here on the assay layout. So now you want to go to data management, delete data, and then you just want to select. So under Dan Spade, I'm going to add my plate name, my measurement name here. So now I can click on Dan, who I'm logged in under. I can see this test folder. I can select everything in that folder that I called. These A is pre-scan, B is rescan. So I had done a couple of those with that had evaluations, and I'm just going to delete it there. So if I'm happy with everything, I have my pre-scan, my rescan, my assay layout, and now I'm going to call my plate name 18 testis. And then you just hit start. And now it's going to go through the pre-scan, it's going to do the 5x, and it's going to go through the 20x on where it found the testis. And then it's going to give you, once it goes to the pre-scan, this max duration time will show up to tell you how long your rescan is going to take, so you know when to come back to grab your samples. And that's that. Obviously, there's a lot of details I didn't go into. You right-click help almost anywhere. It's gonna, the user manual is pretty helpful. And for your specific samples, I can help set up your pre-scan, rescan, and then you can tweak it as needed each time that you go back. So just a quick show. So if you come over here, if you're gonna start, so you images today, you're happy with everything. The next time you come over here, you're just gonna hit this. You're gonna open your pre-scan, go through, test some of your sample, make sure the Z height is still good, make sure everything looks good. And then once you have that and everything looks good here, you're gonna save it, do your rescan here, and everything is already populated. The other way that you can do is go image analysis. You can open your, for example, this is one, a prior one I imaged. I was happy with how it looked. So now you can go back here and it's gonna have your setup saved here as well. And you can see your protocol. You can see which protocol you use for the pre-scan. If you hit load rescan, then this will show you what rescan you used here. And if it shows up gray, that just means there's changes. So you can just look at the name here and then you can load it and start from there. And that is all.